So to begin, we can describe what is multi-omics. Multi-omics, in short, is various measurements of biological data. It contains different layers of different components of a biological context, with analysis can also provide a more detailed view of a given biological context to answer a research question. These different layers, like I mentioned, contain different components of biology. We can have genomics, which would be mutations, copy number, and structural variations, or gene fusions. Epigenomics, which would contain information on methylation and chromatin accessibility. Transcriptomics, which contains messenger and non-coding RNA or transcript fusions. Proteomics, which would contain information about protein abundance, complexes, or possible protein-protein interactions. Metabolomics, which is metabolite abundance. Information about chemistry, more specifically in this context, drug chemical structure, and pharmacology, which would contain information about interest in drug response and cell viability. And like I previously mentioned, using these different layers of multi-omic data really provides an opportunity to have a more integrative, detailed view of a biological context. An example of an application could be in predicting drug response for precision oncology. Let's say we have a cohort of patients, each with the same diagnosis, and we would like to understand a potential drug treatment approach given maybe the presence of a biomarker. If we subject this cohort to sequencing to extract multi-omic information, the analysis of this multi-omic information could provide a predictor of drug response via a biomarker. That would allow us to proceed potentially with treatment and separate this cohort into responders and non-responders. When it comes to the access of multi-omic datasets that you have available for analysis, there has been a steady growth of a number of multi-omic datasets. You can see in this image here, we have the outlining of the development of accessible pharmaco and radiogenomic studies, whose access we'll touch on later in the webinar. You can see the timeline goes from 1997 to 2022, with the number of datasets slowly growing. Not only that, a lot of datasets have an ample number of cell lines and an ample number of different drugs. Datasets like CCLE, CTRP, and GCSI, those are ones I've worked with in my own personal research. And you can see that there's a growing number of datasets available that allow you to potentially answer different research questions surrounding the topic of multi-omic data. Although we have this growing trend of available large volumes of multi-omic data that is ultimately also now becoming commonplace when accessing them through different sources, as well as there's advanced computing and software alongside this generation that is enabling efficient analysis, there are some challenges that exist in the accessing of these multi-omic data sets, both in an academic and a commercial setting. For example, when using these data sets like CCLE, GCSI, 50 to 80% of the time with the raw data object can be spent on cleaning and pre-processing into a suitable format for analysis. Also, the lack of standards can make it difficult to combine multiple data sets from different sources if integration is something that is part of your research. And also, when specifically accessing these data sets, there might be different versions, different contexts, and finding the correct data set for your research can also be very difficult. And so what is the result when we have this trend but faced with these challenges? Well, the result can be slow and expensive progress, suboptimal results, dependent on the available data, inefficient collaboration, or manual repetitive error or being prone to curation operation. So we're faced with these trends and these challenges that is ultimately going to affect the research that is done with multi-omic data. So now I actually want to throw up a poll to understand the topic of multi-omic data. If you work with multi-omic data, what is a challenge you experience? Data with large amounts of noise, extensive pre-processing is required, you have issues integrating data sets across different sources, or you also have issues finding the correct version/contents of a public data set you would like to use. Looks like we have some answers coming in. We'll give it a couple more seconds for you to enter your response. Do so if you haven't already. Okay. Perfect. I think it looks like we have all our answers in. We can end the poll. It looks like a lot of people in the audience resonated with the issues of integrating different data sets across different sources, as well as a lot of people resonated with finding the correct version and contents of the public data set, which I've also experienced when trying to access these data sets. So thank you so much for your input. We'll go ahead and close the poll now. And I'll pass it over to Benjamin to discuss organizing data sets and access methods for future use.
Thank you very much, Emily. So exactly what I expected from the poll. Definitely a lot of challenges in terms of versioning and accessing the right version of the data. So today we'll discuss how we can organize those data sets and standardize them to actually address those changes. So multi-omics is no different than single omics, except that it's more complex. It has multiple layers. So you can imagine that the problem people face with a single single modality of data is actually exacerbated when you deal with multi-omics data. So really the main issue is the lack of standard. You may have samples coming from very, very different sources. They could come from the lab in a cell lines or in a mouse model. It could come from the patients. So obviously, you're going to need a lot of corresponding annotations to understand the origin of this sample. The molecular profiles as well can use very diverse technologies, and they may use very different formats when it comes to releasing the data. And some of those formats might be undocumented depending on the data type. And so the annotations may also not be consistent across data modalities. In biomedicine, we use a lot of the same entities to describe a given sample. So for instance, if you look at molecular profiling, we use genes or genomic regions to annotate the samples, whether it's at the DNA level, RNA level, proteomics level. So we need those annotations to be consistent across the different data modalities. And also we need a lot of metadata regarding how those profiling has been done. What's the experimental design? Do you have replicates? What technology have you used? But also how you process the data. How do you summarize the data? So sometimes you have very complex files such as sequences with billions and billions of very small sequences, and you need to summarize that in a way that you can actually digest for analysis. But the way you do this could actually affect a lot of the downstream analysis. So the use of those metrics for summarization modeling needs to be documented as well. So the crux of the problem now is that most of multi-omic data that are available in the public domain, or you may actually generate within your company, may not directly be amenable to statistical and machine learning analysis. So how do we make those? Can you go back one slide? How do we make those data fair? And by fair, I mean findable, accessible, interpretable, and reusable. So by findable, you want to make sure that the data you've generated and you want to share with the public or within your company are easy to find. So you need the right metadata, and you ideally need some unique identifiers for that data set so you can actually keep track of the versions. You need this to be accessible. Once people find your data set, how can we download the data to actually do the analysis or mount it in your cloud subscription, whatever process you want to use to access the data. I think the big part of the problem in multi-omics is to make sure that those data are interoperable. So annotations are being standardized, the same ontologies are being used, the data are organized in the same way so that those different layers of data, different modalities are actually in sync with each other. So you don't mistake sample one for sample two in another layer, for instance. And ideally, if you do all those things, those data should be really easy to reuse for multiple analysis. And that's where probably you want to bring some code to the data to make sure that you maximize the value of the data by adding some code that show how to load, how to visualize, how to mine, how to analyze those data. So really the best practices now for multi-omics data is not only to have a very strict and proper data management at the very beginning during the data generation process. So you make sure you annotate all your experiments and all the data layers as much as possible. And you want to make sure that the provenance of the data, which sample is being used from which patients or for which model system, for instance, is properly annotated. So you're completely transparent in the way you actually generated those data, but also process those data for the analysis. And you also need very specific unique identifiers for not only the samples in your data, but also the versioning of the data. So again, data sets tend to evolve over time. You may find errors that have been made, some misannotated sample, for instance, or you may have used a more recent technology. So expect those data to evolve all the time. So you really need to be very strict in terms of versioning and put the right identifiers for all those data points. So in my lab, we've struggled with that issue a lot. We deal with a large portfolio of multi-omics data sets for cancer research. So we decided to really codify this process as much as possible. So on the left side, you see a bunch of users, could be a member of your research group, for instance, and they usually, for their own research project, they need access to specific data sets, whether it's internal or external data sets. So the first aspect is how to find and access those data. So it's the search and request layer. And by request, I mean sometimes we can encode multiple ways to process the data. For instance, RNA-seq, you may use Callisto or the Salmon pipeline or the GATK pipelines. But it's hard to know which one is the best. It really depends on your application. So you can let the user actually select the different processing that they would like to use for their own data. That request go to a data catalog. If the data object already exists, meaning a previous user already requested the exact same data, the user already directly get the digital object identifiers and a link to the data. If that data object does not exist yet, then the request is being sent to an orchestration pipeline where all the omics layers, all the omics data are being processed in a completely transparent way. So all that code is visible to the user to create what we call an integrated data object. And that data object actually includes very high curation. All those annotations are being harmonized, standardized. We also make sure that the different omics layers are in sync. Something as simple as making sure that the samples are in the same order, that the missing values or missing molecular profiles are being handled properly. Once this data object is created, it's being uploaded into a repository. So if you want to make it publicly available, we use Zenodo in the lab, for instance. And that will give you a digital object identifier that you can share with your team members, but you can also share in your publications and your reports. And so these data processing layer and data sharing layers are obviously very important for the whole process. But once you have those objects with the DOI associated, now you can finally do your analysis, whether it's on CodeOcean or your high performance computing or your cloud subscription. And really here, I think the crux is that this process being fully automated, you can easily version every changes in the data. And what we found extremely important is that, as Emily said, we spend 80 to 85% of our time, or 50 to 80% of our time doing data wrangling, trying to prepare the data for the analysis. This process is extremely error prone. So once you codify the whole process and you automate it, then you can actually correct potential errors or improve the annotation or update the annotations easily, because now you can just correct the code, rerun the pipeline, and get the new digital object identifier. So you get this new version. So again, this whole pipeline is very tedious to put in place, but once installed, you don't have to redo it, and you can actually improve over time, which is very, very important for us. As I said, data sets tends to evolve. So if you look at this map of public data sets that Emily presented already, my team actually spent quite a bit of time to go through that process for all the data sets that have been highlighted here. And you can see we focus more on the more recent data set, but we really like this initial data set from the NCI, the NCI 60, because they really probed so many drugs. It's a great resource for research in general. If you're interested in creating more of those data sets, we would be more than happy to work with you on that process. So it's really this collection of data set is continuously evolving as new data set get released in the public domain.
Okay, so thank you so much, Benjamin, for kind of outlining data organization and access and practices for future use. I also now wanted to get into potential methods of exploration for discoveries, focusing on and prioritizing to empower scientists of various domains, and I'll get into kind of what I mean by that. So looking at the power of analyzing multi-omics, once it's in a appropriate data processed object to use for analysis. If you look at ways to analyze one-dimensional versus multi-dimensional data, so single genomics versus multi-omics, the type of results you get can be quite different. Let's say you have a sample of cells that you input into a sequencing machine that will output the results corresponding to one modality, maybe gene expression. The results and figures that you will get will be representative of information of just that one modality. It might be a UMAP or TSNE plot representative of cell type, cell cluster based on gene expression, or a heat map of gene expression by cell type or cell group. Versus if you use a sample of cells on a sequencing machine to output multiple modalities, or you use multiple machines to output multiple modalities, the questions can become more complex. You might be able to construct similarity networks, integrative networks that will represent information from those multiple sources. Also, as you continue to use more multidimensional data, it gives you the opportunity to explore questions that might be a little bit harder to validate experimentally, which would introduce the use of AI and machine learning in multi-omic data. And as you can see from this Nature Reviews paper available, there's been a growing interest in developing multi-omic methods, in this case, single cell, spatial, and maybe some other information like proteomic data. From 2015 to 2022, you can see there's been an increasing number of methods becoming available to explore different applications, some of them employing machine learning, some of them not. And they are using at least two or more different modalities from genomic, epigenomic, transcriptomic, and proteomic. And as we continue to move forward in this field, there will undoubtedly be more methods and applications developed, potentially answering more complex questions with more complex methods, seeing the kind of development and rise that AI and machine learning are taking in biological applications. So while these methods and application development is really exciting and it provides an opportunity to explore different ways to analyze multi-omic data, it also is important to understand how this could potentially affect or contribute to a team dynamic. So in computational biology, you may have different team members who have different areas of expertise. An example, kind of the triangle you can see here, is the bioinformatician, the data manager, and the clinician. Each of them may have different levels of experience when it comes to analysis and different results they would like to see. And if there's not a cohesive way of sharing results or providing analysis results between these team members, you can kind of see these speech bubbles pop up that represent a back and forth of changing the results to best reflect the interest. So maybe thank you for sending this result. Can we also look at this expression of this gene in this multi-omic context? Or which source data are we using? Doesn't look right. I think we need to change the input. How did you get the results from a couple months ago in October? I think we should go back to those, but I don't remember what data was used. Or I like this result, but maybe this paper did this. Maybe we should follow the similar trend. So we see these challenges that could arise just from the sharing of the results from multi-omic computational applications. A kind of subset of that challenge in workflows could be centered around visualization, because in representing in a scientific publication, that might often be the method of communicating the results and the efficacy of the method developed. So an example of visualizations that would limit biologists or limit those with less analysis experience would be a static PNG PDF of a result file. It could cause a constant back and forth like we just saw, and there might be a large reliance on the computational biologist, the bioinformatician, the one doing all the analysis to help format scientific discoveries, change the gene expression, change the representation to best reflect a relevant result from the advice of maybe a supervisor or a clinician. Versus we have an option to, instead of limiting, empowering biologists. This can be made available through creating interactive dashboards and apps that will allow for tactile data exploration and allow end users to make their own discoveries. So you can see here, we still have an example of a UMAP plot representing expression of a BRCA1 gene, but we also have a drop down that allows users to select maybe different biomarkers and see the different relative levels of expression versus if we went back to the limiting biologist and wanted to have that same changeability, that would require like 5 different PDFs to be sent and 5 different back and forth communications of, okay, can we see ERBB2? Okay, can we see CD14? Things like that. And we have seen some examples of interactive dashboards being made for multi-omic use. So we see an example using the Shiny interface and creating a Shiny application through the publication from 2021 of My Biomics, which is an interactive web application for multi-omics data exploration and integration, providing different visualizations to understand the integration and similarity between transcriptomic, proteomic, and genomic data. And we also see examples in Shiny's Python counterpart, Streamlit. An example of this is from 2022, titled Stargazer, which is a hybrid intelligence platform powered by Streamlit for drug target prioritization and digital drug repositioning. And as you can see, there are similar figures to the one we saw in the static version of exploration, the limiting biologist result. But instead, the majority of these results are interactive. So you can maybe change around some data overview, change around some parameters to get the result that you want without having to edit the original existing workflow and having to contact the original author of this application. And so given the analysis discussion we had and the data discussion we had and the trends that we're observing in multi-omic data, we can now kind of construct an entire framework for multi-omic data. We have the organization practices involving transparent and reproducible curation of multimodal data using established standards that Benjamin laid out. There's also a prioritization of fair sharing of curated data sets, which would be accompanied with computer code for maybe mining and visualization. And then in addition to fair sharing of the computer code, there's also the computational framework itself, which would enable the collaborative analysis of multiple data sets and empower exploration among team members. And so here's an example of the CodeOcean use that you could use for multi-omic analysis. So CodeOcean provides a compute capsule to do your analysis in. A compute capsule can be seen as a containerization of a scientific project. So if you would like to accomplish multi-omic analysis in CodeOcean, it can be done all within our compute capsule. And so in our compute capsule is housed code, data, and environment necessary to generate a required result. And we have certain features that highlight the best practices of analyses that both Benjamin and I laid out for data and analysis. For example, in the code part of our CodeOcean capsule, we have options to create no-code app workstations powered by Shiny and Streamlit that will allow for a one-click launch of an interactive dashboard created to analyze multi-omics. And also for sharing among team members to kind of minimize back and forth communication, we do have a collaborative working system available that allows users to work with an analysis workflow reflective of their user type, as we have both code and no code options for using our compute capsule. When it comes to storing your data in CodeOcean, like Benjamin mentioned, after executing a pipeline to create a process identifiable data object, it can be stored in a CodeOcean library after analysis. And CodeOcean datasets, they allow you to also provide metadata configuration, including detailed information about a data source, as well as CodeOcean datasets. Generated results are provided with provenance, which will allow you to actually trace back to the correct version of the dataset used for a generated result, which could help alleviate the issue of finding the correct version of the dataset that was used to generate a result of interest. Also with the environment kind of plays into the use of the code for an easy configuration and making the code fair. The environment in CodeOcean is configured with a no-code interface for creating a Docker image for our environment via an automated no-code UI. And the environment is preserved with reproducibility via saved build states. The environment in CodeOcean really removes the back and forth of it works on your machine but not mine. And that could very easily affect a multi-omic analysis. So that's been kind of removed with the way the environment is set up in CodeOcean. So if you were wondering, well, what's the best way I could employ all of these practices in my code and my data to really empower my multi-omic analysis and maybe avoid challenges for myself and for future use of these data sets, that kind of solution is available in CodeOcean. So now we have a second poll to just kind of get everyone's thoughts after we've kind of gone over best practices for data and code. So which component do you think is most important to create a robust analysis framework end to end? Is it the transparent, standardized curation and the processing of data, sharing all elements of the framework needed to run, including environment code and data, or creating results for collaboration and exploration by team members via interactive dashboards? We'll go ahead and wait a little while for everyone to answer their answers, and then we'll look at the results.
Looks like most people are more worried about the early step of the process. How do we get the data right?
For sure. Okay, we'll wait a couple more seconds for everyone to input their results. Feel free to do so if you haven't already. Okay, I think we'll go ahead and close it, and we'll take a look at the results. So like Benjamin mentioned, yes, absolutely. It looks like a lot of people are definitely more concerned with the transparent, standardized curation and the processing of data, which definitely is a relevant component to create a robust analysis framework. Okay, we'll go ahead and end the poll, and I will pass it to Benjamin to discuss a case study of multi-omic analysis.
What I found very interesting about the poll is that if you think about it, if you read a report or a scientific publication, the way data are being processed is usually only a couple of lines. Even so, we spend a lot of our time on these very aspects. So there is a bit of a disconnect between the complexity of the task and what we actually, the importance we give it to when it comes to communication with the public or the collaborators. So today I'll discuss a case study, a multi-omics analysis we've done in the lab using those public data set, but also using the CodeOcean platform. And really, the point here was to make sure that all the team members could work on the same code base, on the same data, and really understand how they generated their results. So today I'll discuss our efforts to use multi-omics data for drug classification, and it's an important problem. When a company creates a new compound or a compound is being discovered, let's say in the natural world, very often we know a little bit about the structure. We can do a few experiments, but it's not trivial to infer, identify the mechanism of action. So that's a very lengthy process, and we wanted to see whether we could have a fully data-driven approach for prediction of drug mechanism of action. So in our study, we are actually going to leverage three different omics or data layers, three different modalities. The first one is probably the most obvious one is the chemical structure. Often we know quite a bit about how the drug is being conformed from a chemical perspective, and we have ways to represent that information, that 3D conformation into 2D strings. So we use the SMILES fingerprints, for instance. But we also, labs, I know many labs are being equipped with, have the capacity to basically treat human cells or the species with those drugs and see what happens. And in cancer research, we do a lot of those drug screening. So we have panels of cancer cell lines. We treat them with drugs at various concentrations, and then we observe, we measure the viability of the cells. If all the cells die, it could potentially be a very interesting drug. If none of them, if they proliferate like the control with no drugs, then this drug is doing absolutely nothing to affect cell viability. So we generate those data very, very often in cancer research. And something a little bit more recent, it's called a drug transcriptional perturbation. So the idea is that we can use those molecular profiling at the RNA level and see how our cells adapt under treatment with drugs. And one of the very famous data sets is called the connectivity map, generated by the Broad Institute. And they use microarrays as well as a newer, cheaper assay to basically treat cancer cell lines with dozens of thousands of drugs and see what happens in terms of gene expression after 4, 6, or 24 hours. So very, very interesting source of data when it comes to understand what a drug is actually doing to the cell. As I said, we are leveraging public data sets to do this. And in particular, we wanted to leverage this old but mighty data set from the National Cancer Institute in the US, where they treated 60 cell lines from different cancer types with a massive number of drugs. So they actually have more than 200,000 drugs with measurements, but there are 60,000 drugs with very high quality multiple replicated measurements. So we use that to basically understand how the viability of cells is being affected by drugs at various doses. And we also use this L1000 dataset, this kind of drug transcriptional perturbation dataset. The flagship publication was released in 2017, and basically they had millions of experiments where they took a cell lines and multiple type on multiple doses and treated with over 20,000 drugs. So very, very rich data set when it comes to understanding how drugs affect the messenger RNA, the transcriptional programs of cells. We also added another data source that was not initially in that map, which is all the chemical structures, all those SMILES identifiers for the drugs that kind of describe the 3D conformation of those drugs. So obviously, you can learn a lot from that data alone as well. And so how do we merge those different data? So every data tells you a very, very different story about the drug mechanism of action, how it affects the cell viability, how it affects the gene expression, what's the chemical conformation of the drugs. And what we wanted to understand is how drugs relate to each other. So we had very large collection of drugs. So we had SMILES for more than 1 million compounds. We had drug sensitivity for more than 60,000 compounds. We had drug perturbation, transcription perturbation profile for 20,000 drugs. So we had very large data sets in our hands, and we wanted to understand how drugs relate to each other. Are those drugs behaving the same way? Do they affect the viability the same way? Do they affect the gene expression the same way? Are they similar from a chemical perspective? But because we could do it at 3 different levels, we needed a way to kind of fuse those different layers, different data modalities into a consensus network. So here you can see as a network representation, that's the similarity between every single drug. So a drug is a node. And we basically, this first paper that was published in 2014, basically the nodes were the patients. So they were looking at similarities between patients. Are patients sharing different DNA methylation or mRNA expression? Do they evolve the same? Do they survive as long as other patients in the network? But we decided to take that paradigm upside down, and basically each node, no, it's not a patient, but it's actually a drug. And so every connection between drugs could be supported by one or multiple layers, and that's how you see it's color coded here. You can see that some connections, some drugs are being similar because they have the same chemical structure and because they output the same transcriptional profile. So we basically merge all that information into that consensus similarity network. So now we have this massive network of drugs that we can mine. Next slide. And here the network looks pretty small because it's already at a community level. So each node is actually a community of drugs. It's a subnetwork of drugs that is very, very tightly connected with each other. So each node, and the title on the node is actually one representative drug in that community. So in this network, you can actually see that there are many different drug communities. Some drugs act very, very differently than others. Some are really at the edge of the network, some are more at the center of the network. So if you have a first-in-kind mechanism of action, if you have a new drug that doesn't match anything in the network, it will be at the periphery. If you have a drug that has multiple targets or acts similarly to other known drugs, it will be more at the center of the network. So now I will highlight a few of those drug communities to see whether the actual network fusion made sense from a result perspective. So here you can see a node called dabrafenib, and that cluster is very, very small. It has only 3 drugs, but they're all BRAF V600E mutation inhibitors. So they're very, very specific kinds of inhibitor, and they were very easy to find even without multi-omics because they had a very, very similar structure. So that was acting more of a positive controls, if you wish, something we knew already just looking by the structure. But there are other communities that we could only find by looking at different multi-omics at multi-omics layer. Next. And that's an example for the microtubule inhibitors. They don't have very similar structure, but they affect the cell viability and they affect the expression in a very similar way. So we found very, very famous drugs such as docetaxel and paclitaxel and vincristine, for instance. And they're all either microtubule stabilizer or inhibitors. But these new compounds, these experimental compounds called SB225002, that was not known to be a microtubule inhibitors, even though it clustered very, very closely to all the other microtubule inhibitors. So we basically looked into the literature whether there was any previous evidence of this to be that as a potential mechanism of action. And indeed, we found that paper in 2013 that described this experimental compound as a potential novel microtubule inhibitor, kind of confirming our prediction here. Next. So we also had another cluster of drugs that was more like a positive controls in the sense that they were very known to be all statins drugs. So they were cholesterol lowering drugs. So the three of them, fluvastatin, lovastatin, and simvastatin. And they're very interesting drugs in cancer research, especially in triple-negative breast cancer research, an aggressive type of breast cancer, where Dr. Linda Penn's lab at the Princess Margaret found that clearly demonstrated that those drugs could inhibit cancer, this aggressive form of cancer. So again, more of a positive control, but quite exciting. But we found this drug partenolate that was a bit of a surprise. We didn't expect it to be there. It has a very, very different structure again, but it affects the cancer cell viability and the transcriptional profile the same way. And again, we found this recent paper showing that partenolate is actually an NF-kappa B inhibitors that could reduce inflammation and lower cholesterol. Even so, the data are fairly weak. Our results suggest that this is an area of research, an avenue of research that could be pursued. Next. A bit of a bigger class. We looked at this very large community, and as you can see, they're all small molecules finishing in ib, afatinib, gefitinib, dapatinib, and whatnot. And they're all EGFR and ERBB2 inhibitors. So again, very positive sign for the predictive ability, for the predictive value of this fusion of this multi-omics analysis. But again, we found this ibrutinib, that is Bruton kinase inhibitors. That's not known to work as a EGFR ERBB2 inhibitors. Even so, there are some papers that suggest that this drug could actually be efficient for EGFR mutated tumors. So again, a new avenue of research dictated or actually identified by this multi-omics approach. And so this analysis was not trivial to do. So we needed to annotate the drug exactly the same way across all those different data sets. We had to combine data sets from various sources. And that's really where CodeOcean came into play to kind of allow the team to efficiently collaborate on that, make sure that the way we process the data, the way we annotated the drugs, which version of the data set we use, everything was clearly documented and embedded in the capsule. So we created data assets within the capsules, we put the code, and everything was reproducible. What I found really, there are two aspects that are very important to me when it comes to research, is something that's easy for people to use so that you have a very low barrier of entry. And here, in one click, you can reproduce all the results. So you know that the code works. Doesn't mean it's correct, but at least you know it works. And the other aspect is that you're not locked on the platform. You can easily export all the results. You can export even all the software environment as a Docker container and share it with the public if they want to use another platform. So really, this aspect was very important for us. And we published that paper in Cancer Research a few years ago.
Okay, great. Thanks so much, Benjamin, for kind of highlighting that case study. Very interesting. So I'm now going to go over kind of a summary of what we've covered today surrounding leveraging public multi-omic data sets for computational research. So we first highlighted that multi-omic data can be described as different layers of biological information to gain a better view of a given context. And we also highlighted that this type of context, including information from genomics, transcriptomics, chemistry, and pharmacology, could be useful in applications like predictions surrounding precision oncology and drug response. But we also highlighted that while there's a trend of growing multi-omic data sets and publicly available ones, there are limitations existing in the data, including lack of standards and lack of proper annotation, and as well as we highlighted in the poll, being able to access the correct version or just a version of the data set. And so ultimately, we described that we need to make sure the multi-omic data is fair. This includes practices involving proper management, annotation, and provenance for a given data set. And it also included Benjamin highlighting a pipeline to make a process identifiable data object that could then be stored in a curated data library like CodeOcean for very easy future use. We also touched on methods of analysis that could empower team members for visualization, including interactive apps or different other methods of sharing results. And we also highlighted a case study of being able to repurpose a method using different omics layers and can show different insights in different topics of research. And Benjamin also highlighted how the work was able to be made reproducible and really easy for future reuse using CodeOcean.